Good morning and welcome back. Today I will be talking about aging and how do we address and face it. And so since I'll be talking more so than perhaps any other talk, these some of the topics get close to having medical applications. And so I want to remind you that I'm a PhD, not an MD. I can't give you medical advice. Um, Another thing I want to just for public service announcement is that there's a lot of supplements out there that are not marketed with medical sounding language, but they're not regulated as tightly as pharmaceuticals are. So they'll make medical sounding claims to try to make the science sound established to the layperson. Um, One way to combat this is if someone's trying to convince you of something, ask yourself and ask them, what does that mean? And don't stop asking, what does that mean? Until they can give you an intelligible response that you understand. Because otherwise, either they don't understand it or they might be trying to pull something on you. So, with that said, I encourage you to find a medical professional that you trust and discuss with them before you start any new medications, supplements, and so forth. Now, the idea of the desire to live forever has been around since antiquity. I have an artist depiction of Alexander the Great on the right, meeting the leader of Persia on the left. Or I say that's my right, your left. I have it reversed. My bad. And so their Persian leaders putting a fish into this pool of water that was dead, and it comes back to life. And this is kind of emblematic of the myth of the Fountain of Youth, which exists in like lots of mythological traditions, and even played a role in the era of the conquistadors, where legends from the native peoples of a Fountain of Youth inspired Juan Ponce de Leon, the first governor of Puerto Rico, to explore Florida. And there's a picture of his men exploring Florida and drinking from a fountain. And since they're not around, we can conclude that they did not find the Fountain of Youth unless they disappeared, right, into Shangri-La or something. Um, but with that in mind, let's think about explore lifespan across the animal kingdom before coming back to humans. And, so there's some remarkable animals that actually are functionally immortal. Both um, this one on the left is the Hydra, and it was named after the Greek myth of the Hydra in Heracles, that because the mythological creature, when you would cut its one head off, it would grow three more. And the Hydra can basically regenerate itself pretty much indefinitely. This jellyfish here is another organism that can essentially regenerate itself indefinitely. However, these individuals probably don't actually live forever because they get eaten. There's like lots of competition. They will eat each other. They will get eaten by other things. Now, as we go into the animal kingdom, we have, or into vertebrates specifically, one of the longest lived vertebrates is the Greenland shark. And this guy has a lifespan of 250 to 500 years. Some, I think, have been recorded as living as long as 700 years. These sharks live in the Arctic Ocean near the bottom, and the temperature, of course, is very cold. It's roughly 0 degrees Celsius. The water doesn't freeze because it's under pressure. And they're not, unlike us, they are cold-blooded. So they do not heat themselves. Basically, they also are remarkable. They're pretty large. They're about 10 times our mass. So these facts, these physical facts, tie into our previous lecture on scaling. In fact, and I'll explain how here with this plot, because on the one hand, on the vertical axis, you have kind of a scaling lifetime as a function of temperature, and another one is a function of mass, and in log-log scale. And I'll explain roughly how the logic behind this. So living systems, as we discussed in our second lecture, need to extract energy from the environment to organize their bodies. And so you have to spend energy to do that because of the second law of thermodynamics. Now, usually we will produce heat and other forms of disorder in the environment, but some processes of the chemical change create disorder in the organism itself. So if you have to spend too much energy to create order in your body, you will end up doing more harm than good. So we talked in the lecture on scaling about there being a fixed number of heartbeats per lifetime across a wide range of organisms. You can generalize that notion. Some organisms don't have hearts, but they still need to respirate. So you can think of there's a fixed number of times that you are processing oxygen in your body over a lifespan. And this is per unit mass across living systems. So if you're bigger, you can harness more energy than if you're smaller. Thus, so to live longer, you can slow down the rate of these So in order to increase this lifespan, you can slow down the cycle of respirations. And that will mean that you can move slower, like these sharks are extremely slow moving. And because they live at a lower temperature, you can extend the lifespan. They're also, because they're larger than us, they tend to have a lot longer lifespan as well. So they added mass and became larger. So these two combined mostly explain the difference in lifespan between these sharks and us. So unfortunately, we can't lower our body temperatures down to 32 degrees. And probably don't try to extend your life by making yourself cold. You'll make yourself miserable instead, and possibly not live longer. Also, keep in mind these things with mass are evolutionary based, so they make sense between species, but it's within a species, having more mass could actually be disadvantageous, as I will talk about later. So don't try to eat more food to live longer. So maybe we can get some more insight into lessons for humans by looking into long-lived mammals. And one notable long-lived mammal is this guy, the naked mole rat. This animal is about the same size as a mouse, so you'd expect it to have a similar lifespan. Mice tend to live about 4 years. The longest living naked mole rat is around 37 years, almost 10 times as long. Now, what's unique about these rats is they live in the Horn of Africa underground, and their tunnel system tends to have low oxygen saturation. So if you recall from high school chemistry, the air we're breathing right now, if I don't confuse it, is about 21% oxygen. And these animals live in 2% oxygen. They don't have much in terms of lung. They have very little. Their lungs are actually underdeveloped, and they have very slow metabolic rates. Those parts might not help us live longer, but their cells apparently have some resistance to cancer in that cells in general in the mammal, once they are touching, don't want to grow as much. So if you think about when you cut yourself, your skin cells have to grow to heal the wound. And once they feel the skin cells on the other side of the wound, they stop trying to grow more so that they don't overgrow. These animals, the naked mole rats, have very strong contact inhibition. So when they sense other cells, they absolutely stop growing, even stronger than our own cells do. And this apparently accounts for some of the lower rates of cancer in this animal. Another long-lived mammal is the bowhead whale. Now, we can't make ourselves large like the bowhead whale. It's the second largest whale. Now, I mentioned cancer, and I'll explain later that cancer remains one of the highest causes of death. And so if we could combat cancer, you could live longer, is the thinking. And you would think that if you think of cancer as arising spontaneously in your cells, if you imagine you're flipping a coin, and if it's heads, you get cancer, and if it's tails, you don't. And Now imagine that that coin comes up heads only 1 in 1 billion times. If you flipped 1 billion coins, it becomes very likely that at least one of them is going to come up heads and cancer will occur. And so there's many, many cells in this in the whale. And so you'd think, oh, it should have cancer showing up. But its rating of cancer is surprisingly low because it expresses a particular protein called cold-induced RNA binding protein. What does it do? This protein helps repair the DNA when it gets broken. So you can imagine if you were reading a book that didn't have page numbers and you had to figure out what order to put them in, that's kind of what happens when your DNA has a double-stranded break. The cells have to figure out how to put the DNA back in order, and it's easy to put the pages in the wrong order and create mutations. The cold-induced binding protein basically has the effect of keeping the pages in order so that you get the book, you rebind the book and put it back as it was. So this is one mechanism that may one day help with reducing cancer. Now we can also look to humans. At the longest living human would be Jeanne Calment, who lived from 1875 to 1997 and is still today the longest living human that we have a record for. She became the longest living human in at least 1995 because she's the only human to live past 120 based on what I read. She remained active and independent past 100, so she maintained health for a very, very long period of her life. And the interesting thing is that you can find pictures of her both smoking, and there are also reports that she would have a glass of port after each meal. So seems to fly in the face of some conventional wisdom. Perhaps another fun anecdote is that in 1965, she was 90 years old. She signed an agreement with a lawyer to turn over her residency. The lawyer was 47 in 1965. And so he's thinking, how long is this lady going to be around? Maybe I can sell the apartment. She outlived him. And his family had to keep paying her because, and it just gives you a sense of like how long 120 years is, because from 90 to 122 is almost my entire lifetime. So, and we have a picture of her at her 122nd birthday down here. Nevertheless, toward the end, she had lost sight and hearing for the most part and succumbed to a lot of degradation over age. All right, so they talked about a maximum lifespan. I just want to elaborate that we often hear a lot about life expectancy in humans increasing a lot. And that's life expectancy increases primarily because we've dealt with things like getting eaten by wild animals, or dying from infectious diseases, or dying because you got injured and couldn't feed yourself. So innovations like that have helped push the life expectancy longer by keeping us healthier and keeping us from dying at young ages. Now, for the rest of the talk, I'm going to mainly focus on aging as it pertains to trying to extend lifespan, more so by treating diseases that arise in old age rather than infectious diseases or other factors. And we can see this plot shows how much infant mortality has changed and how sharply it has dropped over the past 100 years. So lots of ancient societies kind of cluster around this infant mortality rate of around 50% of children would die between ages 1 or roughly by age 15, the age of maturity. And so we see that post-industrial revolution and really post-medicine in the early 20th century, this huge drop-off, almost straight down in terms of that. And this is what is driving most of the increase in life expectancy. So I'm showing life expectancy in England and Wales. You have a nice long time series going back to 1700. And so we can see that life expectancy at birth, which is here in the red, increased the most because a lot of the innovations helped protect young children and help them develop. For the oldest among us, here at 70 years old, things did improve. We do benefit from that, but perhaps not as much as the youngest. And you can also see some interesting events that occurred. For example, the 1918 the 1918 Spanish flu, which actually is a misnomer because it apparently arose at an army base in Kansas, but the kill it particularly killed a lot of younger people. And so you see that plus World War I leading to large declines in the mortality. And you can also see that World War II also led to a lot of declines in mortality for...
You mean the opposite, increasing mortality.
They're increasing mortality. Sorry, thank you. Sorry. Declines in lifespan, increase in mortality. Thank you. Here. So, and one curious piece of life expectancy is that women tend to live longer than men. And so we hear this all the time. You think, well, how general is that? So this is the 2023 life expectancy across a lot of countries in the world. And this dotted line, if you can see it, is the equal life expectancy. And we see pretty uniformly every single country is above the dotted line, meaning that women are consistently living longer than men in almost every single society. This is the same if we go back to 2000. And even if we go back to 1980, we can see that there's only a few countries that are even close to this line, and all of the others are showing this. And so you might ask, why do women live longer than men? And I don't think we have this is one of those cases where there's lots of hypotheses that people have, but no one that probably are partial explanations, but we don't have a complete mechanistic explanation. There's a couple biological reasons. One suggestion is that testosterone may be associated with a higher risk of heart attacks and heart disease. Another part is that men generally are physically larger than women. We have more cells and we have more chances for things to go wrong, so that may increase risks of cancer. There are also potential cultural reasons. I find cultural reasons difficult to explain everything, in part because it's so consistent across many different cultures, and so it's hard to say whether all of these cultural values are shared across all of the cultures on Earth. But speaking generally, men are supposed to be, it's more socially acceptable for them to be risk-taking and ambitious, which can lead to problems. Men tend to have higher rates of suicide, and men are typically expected to be more self-reliant, and that may lead to them, say, avoiding doctors. There's a doctor said, it's like, I know it's going to be bad when a man comes into me and says, my wife made me come in about this. All right. Now, so I want just to emphasize, to set up some ways we could extend lifespan, I want to touch on the causes of death that we see. And so there are causes of death here that are due to infectious diseases, but you'll notice at the top, we're going to see cardiovascular diseases and cancer stay consistently at the top across this time frame from 1980 to 2023. And we see other diseases, diabetes is up there, chronic respiratory diseases, pneumonia. These are all things that you may succumb to in old age. As we go forward in time, we see most of the other causes of death, things like HIV/AIDS is going down, a lot of other infectious diseases, with one notable exception that we will see here in a minute, are going down. Right here, you'll see COVID-19 increased a lot. But in just to mention, you notice COVID-19 has significantly decreased in terms of mortality. That does not mean that you shouldn't worry about it and could still affect older people, but we now have much better medical tools to deal with it, and it shows how medicine can help limit mortality. At the same time, cardiovascular diseases and like cancers remaining high are there because they tend to be things that are accumulation over your lifetime. But let's look at some ways that we'll touch on some ways that maybe we can address those. Now, in order to talk about those, we need to talk about aging across your body. And one thing that I want to alert you to is that if you think about your body, you have cells in it that are dying all the time. And you might have heard this statistic that every 7 years you get a new body. That's not quite true because different cells live different lifetimes. So for example, your blood cells, live about 100 days. Your shortest lived cells are actually in your intestinal lining, in your digestive tissues, and those you're replacing, every week or so. But there are other cells like cells in your brain, the cardiomyocytes, which are the cells in your heart that help it beat, cells that the beta cells of your the beta cells of your pancreas are with you for almost your entire life. So some cells are only a few days old, some cells are going to be years old, and some cells will be as old as you are. And so just like our bodies age, as you probably are acutely aware of, or I'm becoming more and more aware of every day, your cells are aging, and your cells age even their parts, the DNA and the proteins age. And so at all scales, aging kind of reflects this competition between consuming energy to keep our bodies organized, so to speak, and the increase in entropy that can occur when we extract energy. At the subcellular level, this might be damage to your DNA, where your DNA bases get mutated, or it could be aggregations of proteins where your proteins start to stick together and not function properly. And these can be addressed in our own cells with DNA repair enzymes, proteins that repair your DNA, or there's other proteins that will degrade ones that don't work. At the cellular level, your cells can become dysregulated and become cancerous. Our bodies have evolved several mechanisms from programmed cell death, also called apoptosis. There's also autophagy, which is you're self-eating. So when those cells die, you can get the contents of the cell and repurpose them to other cells. And senescence, which keeps cells from dividing out of control. And then finally, at the tissue and organ side, you might have damage through traumatic injuries or degenerative disease. And some stem cells, either sometimes in your body, can help replace these. Prosthetics might help you, manage your function without replacing the tissue. And transplants, in some cases, can help if you lose, say, the function of your kidney or your liver, and you are fortunate enough to get a transplant. I want to emphasize that all of our parts of our body are aging kind of at different rates, and you're only as good as your weakest link, because if it doesn't do really you any good if your heart works really well if your liver has cirrhosis, or if your brain is really healthy and your kidneys don't work, you're going to have a bad time. So part of this is it's hard to know in any individual person what that weakest link will be, which is one challenge with treating aging, because you need to kind of treat the entire body. Now, there's some cells, right? We talked about the jellyfish and the hydra are functionally immortal. And so early in the 20th century, it was believed that our cells could essentially live forever. And so people will ask the question, are cells immortal? And so this French surgeon who won a Nobel Prize related to, I believe, trying to do pumps, but turned out the pumps weren't practical, claimed that he had taken chicken cells out of embryonic chicken cells and kept them alive for 20 years, which is much longer than a chicken would, but no one could reproduce his results. Also, incidentally, Alexis Carrel collaborated with the Nazis and was a eugenicist. So um, I don't feel sad at him being proven wrong. Enter Leonard Hayflick, who noticed in the 1950s was growing stem cells in a dish, and because he took detailed notes in his lab notebook, when he redid the assay several times, he noticed, hey, these things grow for a few weeks and they stop growing. They did it again. They noticed the same thing happening. When he looked, he's like, this happens at exactly the same time. Why is it like the exact same number of cell divisions that these cells stop growing? And that's how he discovered these pieces of DNA called telomeres. These telomeres are structural parts of the DNA that are on the caps of chromosomes, and they can get lost over divisions. So if I kept repeating, to understand how things get lost, imagine you are reading a book that's repeated the sentence, all work and no play makes Jack a dull boy. All work and no play makes Jack a dull boy. All work and no play makes Jack a dull boy. And it goes over and over again 10,000 times. You would have a hard time of knowing whether you had read something 100 times or 101 times. Something similar happens with our DNA because the telomeres have repeat sequences of bases like ACTG, ACTG, and every once in a while they will miss one of those repeats in what's called a frame shift error and that will cause the telomeres to shorten unless it gets corrected. If the telomeres become too short, the cells say, hey, I can't divide because my DNA is falling apart. Don't divide. And so they stop over time. Now, why can we, if telomere loss generally happens in our body, you might wonder, how can we why can we reproduce? Because we don't reach sexual maturity until, age 18 or, our mid-teens. So why is that age not being built on generation after generation? And there are mechanisms to preserve the telomeres both in our somatic cells. And so in the somatic cells are the cells in your body, like your skin cells, the ones that are not your gametes, like the sperm and the egg. And this microscopy here that is at the bottom, you'll see this. We have two proteins that are dyed in red and green, and the DNA is in blue. And so in our normal cells, this protein is at the spot of each telomere. That's what that yellow light shows. But in embryonic stem cells, there's actually different mechanisms that work on this. So embryonic stem cells and possibly also our germline cells have mechanisms to preserve the telomeres. And that helps keep those cells healthy for reproduction. Another part that can age within our cells is our mitochondria. So our mitochondria, sometimes are referred to as the powerhouses of the cell. If you recall from an earlier lecture, they reflect a endosymbiotic event where a microorganism ended up living inside one of our cells. So they also have a small piece of DNA inside them. These mitochondria handle a lot of what we call reactive oxygen species. So when they're making energy, they also have the potential for damaging their own DNA. And because their DNA is sequestered from our nucleus, the normal repair mechanisms can't get in there to repair that. So mitochondrial function can be lost over time, as this kind of shows. So in green, these are healthy mitochondria. In orange, these are unhealthy mitochondria. One way the cells try to deal with this is they are able to detect which mitochondria are healthy and which ones are not, because unhealthy mitochondria put proteins on the side outside of them, and the cell can be like, okay, you're not healthy, go over to this side of the cell, or you're healthy, come to this side of the cell. And they will do asymmetric cell division so that healthy mitochondria get concentrated in one cell. So this is one way that one mechanism cells can use to preserve them. obviously, as mitochondria are key to a number of biological functions, including inflammation and DNA mutations and senescence. And this also raises the question, why is mitochondria age not inherited? It's in part because of the asymmetric cell division that I mentioned earlier, and also because the cells can eat the mitochondria that are not can eat mitochondria if they're not functioning well. And so in women who pass on their mitochondria to their children because mitochondria in the sperm get eaten at fertilization, basically evolution has decided that it's too expensive energetically to police the mitochondria in every single cell in your body, but it can make sure that the cells in the oocytes that become eggs have mitochondrial integrity. Furthermore, the process of fertilization and development also puts a selective pressure on healthy mitochondria, because if you don't have them, for example, the fertilized egg might not implant and the embryo would not grow. There's also these the DNA mutations, the mitochondria are two ways we age, but we can also have age based on whether our DNA is turned on or off. So just to recap, we have the same DNA in every cell in our body, but it has many different functions. And those different functions are because the different spots on the DNA are turned on versus off. As we age, we get some pieces of the DNA that turn off. And so we can actually measure which pieces of the DNA that are turned off by sequencing, the same technology we use to determine the human genome. And so that means that we can actually correlate the methylation with age. And so this actually works well across a range of mammals, and we can actually detect the age there. Now, this, I must emphasize, it doesn't tell you what will happen with your own body if you reverse the methylation. So there's a statistical paradox called Simpson's paradox. So this, like the scaling relationships, holds for different organisms. You can explain difference between organisms, but it can't, for example, explain how old a large dog is compared to a small dog, or explain differences in age among humans. So when you see, there's a lot of science saying, oh, we can determine your biological age with this assay. They probably are not yet at the level that they can determine that inside your body. All right, so we've talked about a few mechanisms behind aging. What can be done to treat aging? And I'm going to tell you about a story that shows the challenges of going from an insight into aging to making it into a drug. So if you remember Morley Safer from 60 Minutes, they did a famous segment in 1991 called The French Paradox. And there were researchers in France who noticed that French people had an unusually low rate of cardiac deaths. And of course, cardiovascular disease is a leading cause of death. If we could cause that to go down and replicate it, we could potentially expand lifespan. And this is surprising because French culture is known for having rich food and drinking wine. And so the conclusion people had was red wine reduces the risk of heart disease. And this was a major boon for the French wine industry, especially because they argued that it was specifically French wine that you needed to drink. This caused researchers to search for why this was happening, because they thought it was in the wine, and they settled on this molecule called resveratrol as a possible anti-aging drug. And it's not actually there's some science about why this might work. One, it's an antioxidant. Antioxidants like vitamin E, vitamin A, vitamin C help protect your cells from aging. This particular molecule is found in the skin of grapes, and it keeps them from wrinkling under the sun. Because when the sun hits the grapes, it can cause it can break chemical bonds, basically. These broken chemical bonds are known as reactive oxygen species. Then they have what's called an unpaired electron. Each one of these lines here would be two electrons that are shared halfway between this oxygen and this carbon molecule. When they become unpaired, when they're broken, they become very highly reactive. But what's interesting about the chemical structure of resveratrol, which you can see here, is that you see these double lines. These double lines mean that there's not two electrons, but there's not just two electrons being shared between the atoms, but four in a double bond. And when you put a lot of double bonds together, it actually creates a larger quantum state so that the electrons kind of can move anywhere between this sequence of double bonds, and it has this way of stabilizing unpaired electrons. Thus, resveratrol and other antioxidants can act like a sponge for the reactive oxygen species, and they can be taken outside of your body before they create damage to the DNA, for example. And furthermore, so we have like a chemical mechanism saying, hey, this might be helpful to stop aging. We also have a knowledge that resveratrol helps increase lifespan by triggering a response that's similar to dietary restriction. So we know that reducing the metabolic rate can extend your life. One way to do that is dietary restriction. Of course, no one wants to do this because it sucks to not eat. But if you could emulate the response of not eating, trick your body into thinking it's not eating when you still are eating, maybe you could extend lifespan. And so resveratrol apparently does this by activating a family of proteins in this that simulate not eating called sirtuin. And by activating this series of proteins, it shows that you can extend the lifespan in yeast, nematodes, flies, and mice. For example, this is a survival curve of mice here. And if they're fed a high-calorie diet, so in red, here you have the mice are eating to their heart's content. And then in black, you have a standard diet. And then you're seeing how many of them live after a certain number of weeks. If you put them in a high-calorie diet with resveratrol, apparently they live longer. I'll make a note here because I won't get into the science of another molecule that comes up here called rapamycin that was found in a fungus in tropical regions. This also works. It works through a different signaling pathway, but it also elicits the same response as dietary restriction. Now, what's the problem with resveratrol? Is that one, the original journalism was based on that popularized the paper. That paper was actually based on some faulty readings of the death records. Causes of death records change, how they're coded changes a lot from country to country based on cultural things. And so French doctors, for whatever reason, recorded a lot of incidences of unknown deaths or unknown cardiac deaths. And when you added these together, it changed from the rate being 1/3 of what it was in the United States to being only 30% less than what it was. So it was in line with other European countries as well. So most of the effect went away. Another problem is that resveratrol is not actually that present in wine. It depends from wine to wine whether it has resveratrol in it. And if you want to have resveratrol in your diet, you can drink grape juice, although that's a lot less fun. The other problem with resveratrol is that it can be quickly metabolized. So your body takes it up, but it breaks it down before it can have the desired effects. Nevertheless, there are over 225 clinical trials on resveratrol on clinicaltrials.gov. If you go through and you look at the ones that post their results, they'll say what their endpoints are. They will say resveratrol treated, resveratrol not treated, and you look at the numbers and you're like, those numbers look the same, which is disappointing. But this is not actually, I'm ragging a little bit on, journalists found a story and it was a little bit too good to be true, but you can understand how an editor would want to run with it. and then can be hard to translate something, even when we know the science at each step, it can be hard to finally translate that to humans because we have so many interacting systems within us. So, but maybe there are still some other options out there for treating aging that are, more promising. One that works but is very creepy is called parabiosis. And what does that mean? It means you take blood out of a younger individual and you transfuse it into an older individual. And so they showed that transfusion, there's been a lot of studies showing that if you transfuse plasma from young mice into old mice, it can actually help them regrow blood vessels in their brain. So I'm showing old mouse with old blood on the left and an old mouse with young blood on the right. And you can see there's more contiguity between the blood vessels in the brain. This also has benefits for like the liver and the spinal cord and other aspects of the brain. They thought one target that has not yet been, developed into a drug is a protein called GDF11 that may mimic the effects of parabiosis without having to harvest blood from young people. This might raise a question of like whether this is ethical because you're probably accelerating aging in the young person by taking their blood more because they have to produce more of it than you are reducing aging. So this probably is not scalable. But it didn't stop the researchers from getting really creepy phone calls from Silicon Valley billionaires asking for plasma from young people. All right, well, let's look at something that might be a little more scalable. And to set this up, we'll talk about, body weight. We know body weight and heart disease go together. Body weight and cancer also go together. And for this reason, public health has been focused on combating the obesity epidemic because there's a lot of negative effects associated with aging that are also aggravated by body weight. I mean, we have type 2 diabetes, we have inflammation, we have immune dysregulation. Obviously, another thing that can happen if you are too overweight is you can lose mobility and you can lose muscle, which can be a big problem. There's also stress on the organs, and it also seems to be bad for your brain. So what if we could have a drug that controls body weight here? And to do this, I'm going to have to tell you about a monster, or actually a lizard called the Gila monster. And they're one of the few venomous lizards. Apparently, people say it's like one of the most painful bites you would get. So don't handle these without gloves. But they're remarkable not for their venom here, but because they live in the Sonoran Desert, which is in the southwest United States. You can see this green area here is roughly their range. So northwest Mexico, southwest United States. And this desert is unique because it has 2 rainy seasons. So basically, the lizard will eat during the rainy season and then it won't eat during the dry season. So it has to go months without eating and regulate its body. And how does it do that? They were able to identify these proteins called exendins they happen to be present in the Gila monster venom. They're not the toxins that create the pain, but they're byproducts of hormone that helps regulate their blood sugar levels. It's similar to a human hormone that's called glucagon-like peptide 1. And if you notice those GLP-1, and you've heard about GLP-1 agonists like Ozempic, they're very closely related. And so this man, John Eng, was instrumental at recognizing that this exendins in Gila monster venom were similar to GLP-1 in humans. And he also discovered that the structure of exendin was because it was similar to humans, it would also not get metabolized like GLP-1 is in our own bodies. And so he was able to make a long-acting drug for diabetes called Byetta. And Novo Nordisk was independently trying to emulate GLP-1s, but they hadn't figured out how to make their molecules long-lasting. So let's look at how this happens. So I'm showing you three sequences of these are sequences of amino acids. So you can think of these are like small proteins, short proteins. They're called peptides. And so each letter stands for an amino acid joined together in a chain. And so up top is the human hormone sequence. And the second one is the drug that Novo Nordisk was developing. And so they did you can see these decorations in here. These decorations keep it from being metabolized, go from 2 minutes to 13 hours. It's a big increase. But it still was getting cut by this enzyme called DPP4 here, which caused the metabolism. The Gila monster exendins have a different structure at this part of the hormone that can't be cut. And so John Eng's discovery, together with the work at Novo Nordisk, extended this to make semaglutide, also known as Ozempic. This goes from 13 hours to 1 week. So these changes were, a couple orders of magnitude. This is a couple orders of magnitude longer. And now you have these long-acting drugs that can mimic the effect of being full without having to, being full and satisfied without having to be starving all the time. And so semaglutide, not only stimulates the insulin production over a long period of time, but it also has a bunch of effects in slowing your gastric emptying, so you feel full in your stomach, reducing your appetite in your brain, so you don't want to eat as much, and of course, stimulating insulin secretion. Now, this raises the question, like there's a recent editorial in Nature Biotechnology asking, are GLP-1 agonists the first anti-aging drugs? And I do want to emphasize some of the remarkable effects they have on weight loss is over a course of a year, if you compare treating with Ozempic versus just diet and exercise, it's 15% weight loss in the case of Ozempic versus only 2.5% on placebo. This is similar to what you'd get from bariatric surgery. Bariatric surgery is where they will go in and they'll cut out part of your stomach to make it smaller. And can be risk prone because if you overeat after doing that, it can be lethal. But so you're getting similar results with a drug compared to a very invasive surgery. It also saw a marked decrease in the number of cardiac events over the placebo group. And so this kind of explains why these drugs are expensive and in high demand. I must mention that because of that, people often turn to like off-label stuff. Do not do that because you have no idea what's in there. The pharmaceutical companies, whatever you may think of them, are legally responsible if they put stuff in there that is not supposed to be in there. If you go buy something from some random on the internet, you have no guarantee that it is what's in there unless you're going to do the characterization yourself. So unless you have a lab and the means to do that, you have no idea. Another thing is that these drugs, you're kind of on them forever if you want to keep the weight off. If you go off them, the weight will come back. And there's also the risk of thyroid cancer. So these might be useful. However, talk to your doctor to make sure that, because they are aware of all of what are called contraindications or things that would say, do not do this because it could be medically risky for you. And again, informational purposes only. I am not your doctor. And with the exception of a few of you, I'm not your dad, so I'm not going to. Now, after making my slides, I actually came across this publication. You'll notice the date is 25th of February this week. And so there's an interesting paper in Nature this week that is studying neurogenesis in the brain. And you probably have heard that your neurons in your brain generally do not grow. And that's what we saw in the cell turnover, is that for the most part, the lifetime of those cells was very, very, the turnover rate was very, very long. But what they found here is they're studying cells in the hippocampus, which is the structure in your brain associated with memory. And so in these last two columns, we have Alzheimer's disease, AD, and superagers. These are people who have old but have remarkably good cognitive skills. And so when we look at the genes, we can find groups of the genes that are on in Alzheimer's but off in the superagers, and off in Alzheimer's but on in the superagers. And you can also look at parts of the DNA and see whether they are accessible to proteins or not. And you can see that large portions of the DNA are inaccessible in Alzheimer's disease, but they are accessible in superagers. So this gives some hope that there's more work in this paper. I'm not going to do a full journal club on it because we're already at the end. But there's some hope that we can untangle the molecular networks that are underlying this, and that you could develop a drug that would stimulate this response and hopefully combat dementia and Alzheimer's disease. All right. Now, aging is not all bad. And you might think, you might think that, yeah, exactly, especially considering the alternative. We see that well-being, there's a curve of well-being that's self-reported over time, and it seems like it decreases on this survey that's given to people in Baltimore. So this is a smaller geographic area, but it's useful because it's been done for a very long period of time. They've done the survey since the 1960s, so they have data from people who lived in the late 1880s. And so based on when you were born, if the later you are born, the more your well-being increased. And in all cohorts, you see that the well-being increases over time. And they were actually able to compare this with a national survey that shows the same effect, although that national data doesn't go as far back in time. So that's one benefit, is life satisfaction. Another is that there's a decrease in loneliness on average between ages 60 and 80. And then most of this increase in loneliness here is explained by loss of mobility. So if you're able to maintain your health, there's actually a lot of quality of life to be had in old age. Of course, you can have knowledge and experience accumulation as you get older, and you can enjoy, new hobbies. Of course, these things depend on maintaining health, as I just explained, and also maintaining wealth. And so we do see that in general, you accumulate age on average, and even in median, you accumulate wealth with age. But one of the problems is that it's kind of what in economics, they refer to it as K-shaped. So if you think about it as if you make more money than you spend, you can save and accumulate wealth. If you are not fortunate enough to make more money than you have to spend to stay alive, you end up becoming poor in old age, and that can be a very bad, poverty in old age can be a very bad combination, which is why, we don't have living memory of life before Social Security in the United States. But apparent that this is something that one of the things that made it popular is that it helped people who were not otherwise able to support themselves. All right, so I'll just remind us that we have to confront our own mortality when we face aging, but scientifically we can choose how we face it, and there are some scientific ways that you can. These are very generic pieces of advice, I'll admit, but things like having a positive attitude can shape your perception, but it can also have positive effects on our brain chemistry. Having healthy habits of the right sleep, diet, exercise, and having fulfilling relationships can be helpful. And those relationships can also maintain health because you are less likely to be you're able to make better decisions when you have people who you trust that you can talk to. So with that, I will conclude. I wanted to recognize all the past 5 years of lectures here and look forward to 2027 and say thank you so much for coming. I really appreciate it.